My presentation is about sampling technique of graphene oxide-based nanometal composites and their influence on pathogenic microorganisms. My presentation includes introduction, a brief overview about nanoparticles, why it's important to study, then synthesis of Rgo, silver, copper, and titanium dioxide composites, next sample preparation methods, and a determination of antimicrobial activity, and finally results. Let's start a brief overview about nanomaterials. In recent decades, we know that nanotechnology has become an increasingly important technology for the food industry. The development of novel nanomaterials makes it possible to improve food quality and safety, crop growth, and monitoring environmental conditions. An important field where the use of graphene composites may have great interest is the packaging industry. And the purpose of food packaging is primarily to prevent spoilage and contamination. This increasing risk in food stimulates scientists to develop new antibacterial nanoparticle substances that have no side effects and are easy to implement. Several examples are described in some manuscripts where nanotechnology has helped improve taste, texture, shelf life, nutrient delivery, and overall quality and safety of foods. There is a variety of metal nanoparticles like copper, titanium, and silver, and they are well known for their strong antimicrobial property toward a broad range of microorganisms such as bacteria and fungi. And our work aims to obtain composites containing nanometals and oxides from them and study sampling technique influence on pathogenic microorganisms that cause food spoilage, because we know that a sampling technique is important on the final results. On that slide, you can see the diagram of synthesis of nanoparticles. The nanoparticles we synthesized with dry method. It consists heating geonitrate complex at 120 degrees Celsius in vacuum, and resulting in an exfoliation and formation of Rgo, silver, copper, and titanium dioxide composites. On the next slide, you can see the Rgo silver composite, and it is obtained by two methods in reduction. First, it is reduction of Rgo silver nitrate suspension with ascorbic acid and thermal treatment. And it goes in vacuum, as I told, 120 degrees Celsius. You can see on the picture XRD patterns, and the characteristic peak of Rgo comes at 10.27 degree, and amorphous complex of silver nitrogen complex is obtained by exfoliation. You can see on the picture. On same imaging, the amorphous complex is disintegrated as a result of thermal exfoliation in multiple layers, on which 12 nanometer silver nanoparticles are deposited, and the silver particles are eventually distributed on the entire surface of the sample. On next slide, you can see graphene oxide nanoparticle with copper and copper dioxide. This composite was obtained similarly, and XRD patterns and some images are given on the slide. Comparing some images of silver and copper composites, it is clear that same process of thermal exfoliation of nitrate complexes take place, and the morphology of the layered composites is similar. The copper nitrate complex clearly shows diffraction maximum at 11.3, and vacuum heating thermal exfoliation of this complex produces Rgo copper composite, which also contains copper dioxide, and particle size is calculated from diffractometric data, and it ranges from 3 to 20 nanometer, and also copper nanoparticle is eventually distributed on the sample surface. On the next slide, we can see hydrolysis of geotitan complex, and it is obtained also by thermal exfoliation, which produces Rgo titan dioxide Athanas composite, which is confirmed by comparison with a diffractogram. Expanded peaks of this composite prove that particle size of nanotitanium oxide is small, and in the range is from 5 to 20 nanometers. The structure and morphology of Rgo silver and copper composites is clearly different from titanium dioxide composite, and these two composites are obtained by three composites are obtained by thermal exfoliation of nitrate complexes, and during first two more gases are emitted than during the thermal exfoliation of titan dioxide. In this case, only water is released. In titanium dioxide case, fluffy powder are formed with a bulk density of 27 milligram per milliliter. Titanium dioxide composite powder are denser, and it is possible that partial oxidation of graphene oxide by nitrate ions, nitrogen oxides, and released oxygen occurs when nitrate complexes explode. So the surface area of such composites is larger than in the case titanium dioxide. This was a brief overview of synthesis nanocomposites, which we used to study antibacterial property. Let's talk about, before we start to talk about antimicrobial property, let's talk about sampling preparation method. First, we took nanoparticles in different concentration, and then we homogenized in saline solution. After this step, we input pathogen in the sample, and we placed prepared sample on a shaker incubator. It should be emphasized that common sampling problems in these cases, sample size, if we take the samples in small quantity, and structure, as we see in the previous slides, was different, and also it is important sample homogenization, which impacts on pathogen activity, because if sample is not homogenized well, they are not typically evenly distributed, and it impacts, of course, on pathogen activity. Antibacterial activity of graphene oxide composites were studied against indicator bacterial strains. These microorganisms, Pseudomonas aeruginosa, Bacillus subtilis, Enterococcus fecalis, these microorganisms were selected to understand the behavior of nanohybrids on different microbial structures, such as gram-positive and gram-negative bacteria. The different susceptibilities to nanocomposites may be due to different cell wall structure and the different antibacterial mechanism of different composites. For instance, gram-negative bacteria possess a thin peptidoglycan layer, whereas gram-positive thick, and which is more resistant to nanoparticle ion diffusion. Isolates were incubated with 20-40 microgram per milliliter of nanoparticles and studied against indicator bacterial strains by variable cell count and agar diffusion method. Nanomaterials of silver showed a certain degree of antibacterial activity, as you can see on the plates used in diffusion test, and viable cell counts on the viable cell counts as well. Silver nanoparticles exhibited different antibacterial activity against pathogenic microorganisms, and good antimicrobial effect of Rgo silver nanohybrids could be explained by combined action of direct contact between silver nanoparticle and the microbial cell and dissolution of silver ions from the silver nanoparticles. Results demonstrated that silver nanoparticle fully inhibited gram-negative bacteria and significantly reduced gram-positive microorganisms. As you can see, the next slide, copper nanocomposite showed also good sensitivity toward all microorganisms, but in comparison of silver demonstrated that silver ions have stronger activity than copper. The effect of titan dioxide nanoparticles against gram-positive bacteria was less than gram-negative due to difference in membrane structure of bacteria, as we told, but it was greater than in the case of copper nanoparticle. Titan nanoparticles showed high antibacterial activity, which may explain that crystalline structure and shape of titan dioxide nanoparticles are both considered as the most important condition that affect in antimicrobial activity. We made two different experiments with a sampling preparation method. In the first example, we followed all instruction which is necessary, let's say necessary in sampling preparation, and in second one we didn't homogenize, we did not put the samples on shaker, and final results showed that microorganisms can't interact with nanoparticles as well than in the first case, and inactivation level is too low. The results demonstrated that depending on homogenization of the sample of silver, copper, and titanium dioxide exhibited significant antibacterial activity compared to not homogenized. All nanoparticles fully inhibited the growth of pseudomonobacillus and significantly reduced Enterococcus faecalis growth at 2 and 24-hour in the time-dependent rate compared to the respective time controls. So I would like to thank to the staff of Petromelekishvili Institute of Physical and Organic Chemistry who provided us with graphene composites, which we used to study their antimicrobial activity. Thank you.